at least you'll have a little bit more time, hopefully. Lab quiz shouldn't be that bad. So next week when we come back, we're going to do a lab on Monday, which like I told you guys, it's a cleanup of your drawers and all of that, and then we do the ELISA, which is pretty fast. Then we'll come up and we'll do the nervous system. Now, on that Wednesday, again, we can decide on Monday on that. Like I said, since we don't have a lab, I know some of you guys drive a long way here. If you want, I can do it on Zoom. How many of you guys would like to have it on Zoom? Oh, majority. Okay, so we'll do that on Zoom. Okay, so the Wednesday before Thanksgiving, I'll be on Zoom. I will be here because I have my morning class. So I will come here, morning class, they're all from local areas, so they will come. So I will be here. If you guys want to meet with me or something, let me know. But then I'll start the Zoom probably around 11:30-ish, something like that. So I'll put it on Canvas, the Zoom link and everything. All right. So I wanted to give you guys a heads up for the lab quiz. You've covered everything. You've gone over it. ELISA is not that much more complicated when you need to know about it. So if you want to get a head start for it, you can maybe start studying for the lab quiz now, this weekend that is coming up, and then leave the most of the other study time for your lecture exam. Okay, lab quiz. The setup, and I'm recording this, so I'll put it in an announcement on the Canvas separate from the lecture recording. So the lab quiz is going to be pretty much the same format as the lab quiz 1 that you had. So short answers, fill in the blanks, complete the table and things like that. On it, since you guys did unknown, there is some of the assays, gram staining, that is basically filtering it into quiz 2. So gram stain, same things that you guys learned about for the first practical and first quiz. Do the same thing again, study those, be able to again identify gram positive, gram negative images if we have it on slides. Same thing with SNOT test. And then the other thing that comes here is the exoenzymes and the, we didn't do lipase, so I gotta modify this. I have tributyrin, which you guys didn't do. I know I removed it from the first, but I forgot to remove it from here. So exoenzymes, like the starch, some of you guys did that for your unknowns. Gelatinase, casease, again, same thing, urea. So same as you studied before. What is positive, what is negative, and again, if I show it to you like urea, this is control, this is a pink broth. What does this mean? This means it's urease positive. It is capable of producing urea. Things like that for the practical. So I'm going for both. On the quiz, obviously you don't have images, but you know, I can say what is for a urease positive test, how does it, what indicates that the urease is positive? Appearance of a hot pink color, something like that. Let me do this right here before I forget. And I'll upload the updated one there. Selective differentials. Most of the ones that you did, like mannitol salt agar, EMB, you guys did those. So make sure that, again, you kind of look at that. EMB and McConkey are very close. Some of you guys did the triple sugar iron TSI. So have an idea about those. Those were covered on the quiz. Here, these are new. Kirby-Bauer. Have an idea of what is a Kirby-Bauer, disc diffusion. So you basically, the process is called Kirby-Bauer, or susceptibility disc diffusion test. The principle behind it is you have discs that are filled with antibiotics, and then you place it on a plate that has been inoculated with a specific bacteria. What is a zone of inhibition? And then understand that zone of inhibition here, you have to just measure it in order to be able to figure out whether it's susceptible. So what are the terms susceptible, resistant, intermediate mean? Know that. Then we did the INVIC. So again, same thing. Understand what are the medias that we use for indole, for example. Indole production looks for the enzyme tryptophanase. So again, if there's an enzyme in there that you're looking for, what is the name of the enzyme? What is the substrate? So for SIM and indole production, we're looking for the enzyme tryptophanase that breaks down tryptophan and produces indole. So the substrate for it is not indole. Indole is product. The enzyme is tryptophanase, and the substrate for it is tryptophan. Okay. SIM-agar also does H2S production. Citrase. So again, you know, same old, same old. These are know what is positive, what is negative, how do you read it. This one you have to add coax reagent. Citrase is Simmons. You can actually see it clearly. You don't need to add anything. Methyl red, now BP, what is the principles behind it? Some other endoenzymes that we looked at. H2S production, catalase, oxidase. And this is from chapter 15. So we obviously did two things also here. PCR, mini PCR that we did, and we're going to do the ELISA. So the PCR that we did is the genotypic method of recognition. What you need to know about PCR is, first of all, what is PCR? Polymerase chain reaction, artificial DNA production in a laboratory. What is a Taq polymerase? How do you separate the strands of DNA? Using heat. So some of those principles of PCR know it. Then as far as the rapid PCR, again, understand that it's rapid because you now have added a dye that actually can show you when DNA is made and amplified. So you don't actually have to run a gel or anything. Because you have that dye, you can see it rapidly. As far as the COVID went, we can use it even for RNA viruses. Why? Because we have reverse transcriptase. So reverse transcriptase, know what that enzyme is. It's an enzyme that takes a single RNA strand and makes a double-stranded DNA from it. Okay. So I have an idea for those. Okay. And ELISA, we're going to do it. ELISA is an immunoassay. Now that you've done your immunology, you know that for any antigen, we produce antibodies. And antibodies attach to their specific antigen. That reaction, we can actually use it for detection. So and we'll talk about that on Monday when I go over the introduction for it. So we can actually use that antibody-antigen interaction for multiple things, like figuring out whether or not a person was at some point exposed to a pathogen. If you have antibodies in their system, this person produced antibodies, so they have been exposed. You can use it for diagnosis by using antibodies, take samples from the patient and see what antibody binds to this antigen that they have in their sample. So things like that. And we'll go over that. And I'll mention to you guys again key things that you need to know. So the study guides is pretty comprehensive. It's pretty good for you to study it and learn from it. Okay? So that quiz is after we come back from Thanksgiving. All right? Questions for me? Okay, so I'm going to stop this recording. I'll put it in the announcement as far as the how to study for the quiz, the recording for that.